Good day everyone, I am Ramelin S. Sadlaon, and today I will be discussing the DNA structure and the DNA replication. We will go first with DNA structure. DNA stands for deoxyribonucleic acid. It is mainly composed of three components, which are the phosphate group, pentose sugar, and the nitrogenous bases. There are four nitrogenous bases found in DNA: the adenine, guanine, thymine, and cytosine. These nitrogenous bases can be categorized into two, namely purines and pyrimidines. Adenine and guanine are examples of purine bases, while thymine and cytosine are examples of pyrimidines. Moving on, each component of the nucleotide is held by a linkage band. These bands are the phosphodiester band, glycosidic band, and the hydrogen band. The phosphodiester band is the linkage between the 3' carbon sugar and the 5' of the other carbon sugar. Glycosidic band, on the other hand, links the sugar and the nitrogenous bases, while the hydrogen band is the band between the nitrogenous bases. In pairing these nitrogenous bases, purines should always be paired with pyrimidines. So, purine adenine should always be paired with pyrimidine thymine, and purine guanine should always be paired with pyrimidine cytosine. Adenine and thymine are tightly linked with two hydrogen bands, while guanine and cytosine are linked by three hydrogen bands. A and T bands have weaker link than G and C bands. We all know that DNA is double-stranded and it forms a helix structure. It is composed of two linear strands that runs opposite to each other, which means they run in opposite direction. One linear strand runs from 5' to 3', and the other strand runs from 3' to 5'. Their direction is anti-parallel with each other. The 5' to 3' is what we call the coding strand, and the 3' to 5' is the template strand. We are finished with the DNA structure, so now we will be moving on to how this genetic material replicates. DNA replication occurs in the S phase of the cell cycle. It is the process where two identical replicates of DNA are produced from one original DNA molecule. It occurs in all living organisms, and it is important because it acts as most essential part for biological inheritance. In eukaryotes, including us humans, DNA replication occurs inside the nucleus. For prokaryotes, because they don't have true nucleus, it occurs in the cytoplasm. Now we will discuss the steps of DNA replication. The first step in DNA replication is the annexation process, where the annexation protein binds at the origin of replication, creating tension in the adjacent nucleotide. The tension now disrupts the hydrogen band between the base pair that makes the DNA molecule to slightly open up. The second step in DNA replication is the unwinding of the DNA double helix. The initial break in annexation process provides access to the helicase to further unwind and separate the DNA double helix by breaking the hydrogen bonds between each base pair. The DNA helicase binds to the lagging strand template at each replication fork and moves in the 5' to 3' direction along with this strand, breaking the hydrogen bands and moving the replication fork. As the hydrogen band breaks, it exposes the single-stranded DNA. The exposed single-stranded DNA is unstable and they are prone to reannealing and degradation caused by nucleases, but the single-stranded binding protein helps prevent this to happen, and it stabilizes the single-stranded DNA structure. As the helicase further unwinds the double helix, torsional tension will start to build up ahead of the replication fork. Unable to avoid this, the enzyme topoisomerase or DNA gyrase helps control and reduces the supercoiling and the tension. The third step in DNA replication is the elongation. It involves most of the enzymes. During elongation, the enzyme primase synthesizes a short stretch of RNA strand or the primer to provide 3' OH group for the attachment of the DNA nucleotides to start the elongation process. DNA polymerase attaches to the 5' phosphate group of the first succeeding DNTP into the 3' OH group of the primer or the last nucleotide in the growing strand. This process simultaneously takes place in both strands. The elongation process produces a leading strand and a lagging strand, where the leading strand spends less energy as it only needs one primer to complete the replication process. Lagging strand, on the other hand, needs more primers as it goes against the movement of the replication fork. Primer removal will follow. The primers in both lagging and leading strands are removed and it is replaced with DNA nucleotides by a specialized DNA polymerase. The removal of primers along the lagging strands exposes the stretch of unconnected DNA fragments known as Okazaki fragment. As the DNA polymerase replaced the primer molecule with free DNTPs, it still has a gap, but it will be sealed by the enzyme DNA ligase that creates a phosphodiester band between the 5' phosphate group of the first nucleotide to the 3' OH group of the last nucleotides of two adjacent Okazaki fragments or newly formed strands. The last step in DNA replication is the termination process. There are two types of termination process. First, when two replication fork meets. Second, when there's a presence of termination protein. As the two replication fork meets, they will fuse the DNA and two identical DNA molecules will form. So that's it for today. I hope you learned something. Thank you.